into modules, go to attendance. I hope I activated today's. Yeah, go to November 10th. Go ahead and click on submit assignment. All right, yesterday we started talking about DNA.
You said press the announcement?
No, attendance. And we're fine, go to modules, attendance. And every day you have an attendance assignment to do. It's like a warm up. All right, yesterday we were talking about DNA and RNA, and so here we're going to do look at similarities and differences. So for DNA, what is you make a I don't think you can make a chart on there, but you could do. All right, so we want differences. What DNA? What does it look like?
X, Y, Z.
No. How do we describe DNA?
So what it look like? You feel like X.
What does DNA look like? Or how would we describe its molecule? A ladder that's twisted. So we call that a double helix. All right, what else do we know about DNA? What else do we know about DNA? If you call up the notes from yesterday, that I put in there, what else do we know about DNA?
I got 12.
The very end was a chart. Supposed to fit. Oh, no. Right here. So, double helix. What else? What type of sugar does it have? It's a pentose sugar, but the name of it is deoxyribose. And then it has bases. Look at the bases. A, T, C, and G. So RNA, instead of being double, it is single. It has a ribose sugar, so the sugar is different. And the bases are the same except for right here. RNA has U, DNA has T. So those are differences. So you could type it out, DNA, or an arrow or hyphen, and then list these, comma, comma, comma. You can do RNA, hyphen, list those with a comma for number 1. And I guess for here, you could say it has, because we're doing differences, a T base, and this one has a, so these are differences. Number 2 is similarities, 2 things that are similar. So they both, so this is for both, have, and the bases that I erased were A, C, and G bases. And then when we talked about the nucleotide, so they're both made up of the monomer, which is a nucleotide. All right, so 1, you're listing the differences, and 2, here's the similarities. Once you get those typed and submitted, then I'm going to have something for you.
Alex. Let's make Kansas as bad as possible. Want to? See if we can make a swing. That's fucked up. Chicken nuggets.
All right, everybody have submitted. All right, then you're going to go to modules. And you are going to come down to,
Yes, sir.
that's enough, guys. You're going to go ahead and look at the video protein synthesis while I am getting the copies I need. Most of what you're going to do today will be on the paper, so you need to watch this video. All right. Go down and get to the video. All right, then number 2 is right up there. Boston. Press enter and do number 2. Certainly on the board. The video is protein synthesis. Does it say protein synthesis or did I choose the wrong one? Under Tuesday, protein synthesis. Go down
What?
Bro, y'all, I went to the freaking, what's his name? I went to a party, bro.
Dude, I like it. I like that they were metal jewelry and metal belts.
But it was $11. I had it yesterday. You know what Dollar General is?
Where between your place.
The one by the car wash. That's
Where
where
your
I got
place
it.
is like, oh my bad. Walk again, and I wind it up. Then I walk. We were.
The dude, he real cool. I was hot.
Walk in, slide it.
This shit,
To the left side, you shoot it. They give you a special thing.
I don't go to that one.
Okay, listen to the videos.
There's one right next to the food line, closest food line. I'm going base boosting now. Oh,
All right, if you will pause the video, we're going to do something a little bit different today. You have a note packet on your desk so you can put your Chromebook to the side.
oh, we finally get paperwork. Thank God. I'm so tired. I never wanted to do paperwork. Man, back in my life.
Yeah, give me just a minute. All right. So you're going to fill in as we go through this. We're going to be talking about protein synthesis. Proteins are micromolecules that are within our bodies that allow us pretty much to function. We need proteins, and synthesis is to make. So this is how proteins are going to be made, is the process we're going to look at. And so they kind of have something called a central dogma of genetics, and pretty much it goes DNA, RNA, protein. So you have to have the DNA to get the instructions. They make a copy of the instructions and put it in the form of RNA. And then RNA is the instructions because it's small enough to get out of the nucleus and go to a ribosome to make the protein. And so we're going to look at the two steps of transcription and translation. And then we're going to be practicing that as well. So this is just the process of making DNA. Did everybody get those blanks filled in? It's word for word. Reading the instructions in the DNA to make protein. DNA is instructions. If I make it large, it will not video. All right, so we're going to look at the types of RNA that's involved. So the next section deals with the types of RNA. Number one is mRNA. The M stands for messenger. It is what is going to come from the nucleus. It has a copy of the instructions, and it's what's going to go to the ribosome, and it has the directions for making that protein. You have a tRNA. The T stands for transfer. This is the second type. It's going to carry amino acids. Amino acids are the building block of proteins. It's going to carry the amino acids to the ribosome, and it's going to pair up to the message and deposit the amino acid. Then you have rRNA, which is ribosomal RNA, and it composes or makes up the ribosome. So three types: mRNA, tRNA, and rRNA. And they each have a purpose in protein synthesis. Remember, RNA is single-stranded, contains ribose sugar. It will not contain thymine or the T base. They will contain the U base. Stop with the noises. All right, so we're going to look at transcription. Transcription. So the purpose. So you have a place to write the purpose out. This is to get the instructions out of the nucleus. That is the purpose of transcription. Transcription. Script means to write or copy. So DNA, the molecules of DNA cannot leave the nucleus. They are too big. And so each chromosome has a long string of DNA, and on those strings of DNA are genes, and only certain genes will code for certain proteins. So the copy or the transcription of that specific gene is what is going to be put on a messenger RNA so it can leave the leave the nucleus. Please keep your mask on. Mask on, please. It doesn't matter. No. Location. This is happening in the nucleus. It's happening in the nucleus. It's going to start with the DNA, and then it's going to end up with your messenger RNA. So transcription is getting a copy of the instructions in the nucleus. It's getting it from the DNA to an mRNA, messenger RNA, and the messenger RNA is what is going to be moved or be able to leave the nucleus to go to a ribosome. All right, so kind of the process of what's happening. Number one, wherever the gene is located on the DNA that needs to be copied, that piece of the DNA needs to be unzipped. So DNA has to be unzipped. So number one is unzipped. you're going to use complementary base pairing rules to match RNA. So that first blank in number 2 is complementary. And then the second blank is match RNA nucleotides to what is exposed of the DNA nucleotides. But RNA, when it comes to an A on the DNA, it will put a U on the RNA strand. It doesn't use thymine. A will bind with U. Once the RNA molecule is made, it's released. So number 3, release the completed, and then mRNA. So number 3, first blank is released. Second is mRNA. And then 4, the DNA zips back up, and the mRNA in that blank is leaves the nucleus and enters the cytoplasm. So all that's happening is transcription as this messenger RNA is getting built, and it's a copy of what's in the DNA using these complementary base pairs. So that is transcription. Now, if you look at the bottom here, we're going to do this little copy in just a minute, or this example. Let's see. Everybody get those? All right, so here's another picture. You have DNA. The DNA splits. You have the RNA being built. Notice there's no orange pieces now. They are gray because they're U. The DNA comes back together, and you have your mRNA that will then be able to leave the nucleus. So, because I can't do it in the present mode, so if you notice here, wherever I have a T, I still put a A. But wherever there's an A for the RNA, I will put a U. Anytime there's a C, it pairs up with a G, G to C, C to G, T still pairs with A, but A will pair with U. And so that's how that goes along on that example. So what on a test or quiz, you may want to scratch sheet of paper, and you may want to write down for DNA replication, it's this, but for protein synthesis, stop whistling, A will pair with U, and C will pair with G. So it's a little bit different. All right, once it gets out into the cytoplasm, we have some words or some terms that you need to be familiar with. Genetic code. So that's the next part. Genetic code. It is the code of instructions for how to make proteins. So instructions and make. A codon is a set of 3, and you may want to draw the arrow on your notes so that you know what I'm meaning by codon. It's a set of 3 nucleotides. It is read in groups of 3. Groups of 3 make a word. So a codon is like a word. It's a group of 3 nucleotides. So a set of 3 goes in those blanks. And you're going to ultimately make an amino acid, which is a monomer, first blank, for making proteins, second blank, held together by peptide bonds, third blank. And then you have the term anticodon. So think of anti as against, or I think of it kind of as opposite. So it's going to be the matching pairs for the codon. They're found on the tRNA. So it's complementary is the first blank. 3 nucleotides on tRNA. So these are the terms you're going to hear when we're talking about translation. So translation, if I want to translate something, I am trying to put it into a language that I understand. So we're taking the genetic code here and we're turning it into something that the cell understands or the ribosome understands so that it can make the designated protein. So we're going to look at the purpose of translation. So it's to read or follow the directions on the mRNA to make, huh, 1 second. Got it? All right, so the purpose of translation, to read or follow the instructions on the messenger RNA to make a protein. This happens in the cytoplasm at a ribosome. Ribosomes are either floating in the cytoplasm or they're attached to the endoplasmic reticulum. So location, cytoplasm at a ribosome. Yeah. It's going to start with the messenger RNA, and when it goes through this whole process, you're going to have a protein. So again, the purpose is to read or follow the instructions that's on the messenger RNA to make a protein. It's located in the cytoplasm at a ribosome. It starts with the messenger RNA, and it will end with the protein. End with the protein. All right, now we're going to look at the process. Sort of the step-by-step process here. So it's going to start with the messenger RNA. Will attach, so the first blank is attach to a ribosome. Second blank, ribosome. The ribosome is going to read, number 2, the first blank is reads, the mRNA. The second blank in number 2 is codons. Remember, that's 3 RNA nucleotides grouped together. Number 3, you've got tRNA in that first blank. That's transfer RNA. Molecules will act like taxis to pick up and drop off the amino acids is the next 2 blanks, amino and then acids that match the current codon. 4, the tRNAs continue to drop off amino acids. The ribosomes bind the amino acids together with peptide, is that blank, bonds. Think of proteins, peptide. Both start with a P. You need to know that those are the bonds that are in protein. 4, the blank is peptide. 5, when the stop, first blank, stop, codon is reached, the ribosome releases, is the next blank, the completed protein. Now, this was, I guess it was back in the 90s when they actually decoded our genetic code. So scientists finally figured out the code, figured out the bases, and came up with a code. And so we're going to learn how to decipher some of this. So here's a picture of kind of what it looks like, translation, sort of in a graphic or a visual. So it's going to be interpreting, this is translation, it's interpreting what is on the messenger RNA, turning it into a protein. These funny little things are TNA, looks like sorry pieces in the sorry game. They're going to connect these circles up here, which are amino acids with peptide bonds, and you're going to have your protein in the end. Now, yeah. So here's our wheel. There's two ways of charts, and I'm going to have a printout in just a few minutes where you have one of each that you can use. You always start in the center. So if I were given, you always have to have messenger RNA. If I were given G, G, G. All right. If you were given something like this, the first thing I would do is I would go to every three and draw a slash to separate them. Then to use this chart, I'm going to go to the center for the first letter. So I'm going to go to the A. I'm going to go to the U in the next rung out. So still touching the A. Here's the U that touches that A. And the last one is G. I'm going to go to the G. And now I see the amino acid. We usually use the first three letters for that. So that amino acid would be MET. Please stop. Go to the middle again, the next three. You have a C. So I'm going to go to the C. You'll go to the next one, A, and then you're going to go to the U, and that will be his. I'm doing an example that's not on your paper showing you how to read the chart. All right, so the next one, you have a G. I'm going to start in the center for the first letter. Going to go to the next rung, G, and it happens to be a G. This time it is GLY. We use the first three letters typically. You have decoded that messenger RNA strand into the proteins for guys. And this is what we're going to be doing some practice. So if you will look right here. I'm going to present it. This is what happens. See if it still records or cuts me out. All right, so here's your DNA. First thing you have to do is turn it into RNA. So if I wanted to turn this into RNA, what would I wanted to do? Not what I wanted to do. I want to do this one. All right, so if I wanted to, all right, so first you have to get the mRNA. So on your paper, let me write with my finger. Supposed to let me write with my finger. All right, if I want the mRNA, what will T pair up with?
Hey, wrong.
A, yes. And I like to do a little line. A will pair up with, if I'm doing mRNA,
AUG.
this is going to be U. This will be G. G goes with C. C goes with G. T will go with A. This is U, C. So this is the first step. A, U, G, C, U, A, A. Now, what I like to do, put your mask on, please. I like to go ahead and separate them out into groups of 3. Then you're going to use this little chart that's on your paper here, and you're going to find um, these are the codons, so you don't have to rewrite it. So you're going to find the amino acids. So I'm going to go A. So I'm going to go to A in the middle of my chart. I'm going to go out to the next rung, which is U. And then I'm going to go to the third rung out, which is G. And then I'm going to see the term, the word that's there. I'm not going to write that whole word. I'm just going to use the first 3 letters. So I'm going to put met for this first one. A lot of times we do hyphen. All right, the next one, C. So I go in the middle, C. Then I'm going to go to the next rung, G. And I'm going to go to where A is. And it looks like all of them are in the same, so it's ARG. So I'm going to put ARG for amino acids. And on yours, remember there's a spot for codons. I separated the codons with the lines, so you don't have to write anything there. The example on the bottom of 10. So we translated to mRNA first. We're skipping the codons because that's my little lines that separated groups of 3. Amino acids is what we're doing. So I'm on the third one, so I'm going to go to U in the middle. I'm going to go out to the C, and then out to the A, and that is SER. So SER will be next. UGC is next, so I go to the middle, U, I find G, I move out to C, and it is CYS. And then the last one, U in the middle, A, and go out to where you see the A in the next one, and it says stop. You don't write anything for codons, Henry. So UAA is stop. So that means that it will tell the ribosome that it stops coding and releases the protein. And we're going to do some practice of that in just a few minutes. We have one little chart left to fill in on the back. Has everybody got this so I can get off of this right here? And I want to escape. And okay, so I guess the chart is not there. Huh? You're always going to start in the middle. So A, go to the next row in this top piece, which is U. And find G right here. And then you press your buttons. All right, so let's fill in this chart that is not on my thing up here. All right, the purpose of DNA replication is to what? To make 2, what would we we put in that blank? If we're gonna replicate something, we want what? Identical copies. So under DNA replication, purpose, make two identical copies. Those two blanks
Like what?
in the chart. Underneath it, it says nucleotides. The nucleotides for this is A, C, D, G. I don't care what order they are. Where does this happen in the cell? It happens in the nucleus. Transcription is next. And it's to produce a strand of, the first blank is mRNA from DNA, nucleotides, A, U, C, G. Where does this happen? Nucleus. It involves DNA, it's going to be in the nucleus. Then for translation, it's going to produce a protein from messenger RNA. You will need tRNAs and amino acids. I'll move in a minute so those behind me can see. And this is going to happen in the cytoplasm at a ribosome. I'll move. All right, so the last thing that we're going to do is look at this right here. Let me get this. All right, let me get this up in a, yes.
I like your.
Let me get this up and I'll do one example and then everybody's working. All right, I'm gonna put this where I can write on it up there. We're gonna go through one example up here, and then you're gonna have three you're gonna work on your own. Oh, my Jamboard, where did it go? little wheel. And there's 4 problems. We're going to do the first one together. All right, so it's going to be a little blurry, but we can get the job done. All right, so for mRNA, you need to use A. Anytime you see an A, you're going to put an E. A U. This is DNA. When you see a T, you'll put an A. When you see a C, you'll put a G. When you see a G, you'll put a C. If it helps, write that down. This is your DNA. This is your mRNA. So here I have a T. So I like to draw a little line so I don't get confused. That's going to be A. That's going to be U, G, G, C, C, A. I'm going to put U, T, A. Oops, that's T, A. There's U, not T. U. That's why I like to have this. I even will get going and mess up. A, D, U, 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 A, and G. All right, before I do anything else, I like to go through and mark them off in groups of 3. Then you're going to use the wheel to find the amino acids. So I'm going to get you started on this. So A, we should already know what AUG is now. We've used it about 3 times. Go to the A, go out 1, to the U, find the G, and that same little copies is MET. So you're going to write MET hyphen. You're going to go to the next one, G, C, C. G, C, C, A, L, A. And you're going to continue until you fill that in. So for the remainder of class, you are completing the rest of 1, 2, and 3, and I will collect this at the end. I will collect this at the end. As soon as you finish number 1. Guys, do you need the bathroom? Do you need the bathroom?
Yeah.
I'll put this back up in a minute. Doctor's note is there.